Hello and welcome. This is our presentation for Assessment 1, Constructing a Phylogenetic Tree using Mega 11. Step 1. Align the DNA sequences in Mega 11. Start by clicking Align, then select Build Alignment. Choose Create a new alignment and click OK. When prompted to choose between DNA, select DNA. Next, delete the default sequence by selecting it and clicking Delete. Then, click Insert Sequence from File and locate dataset 2 in Mega X. After importing the sequences, align them using the cluster W method and confirm by clicking OK.
Then, you will show the cluster W parameter. Click OK and wait for the alignment by cluster W.
When the alignment is complete, check that all sequences are present and none are left to delete. Save the file as dataset2 alignment. Click on the Model menu, select Find Best DNA Models, Maximum Likelihood. In the Document section of Mega, open Dataset2 Alignment and click OK to begin.
And for the confirmation of the protein coding nucleotide sequence data, just click No before clicking OK after checking the Mega Analysis preference and wait for the progress.
Mega will suggest the top model. For example, T92 plus G plus I.
Lowest model is K2 plus G plus I with parameter of 24. Next, we will show how to construct a phylogenetic tree.
In Mega 11, go to the Phylogeny menu, then select Construct or Test Maximum Likelihood Tree. Click Yes to continue. In the Model and Method box, select the Tamura tree parameter model and click OK to begin tree construction.
And after waiting for the progress, the Mega 11 will generate your phylogenetic tree. Click Save under the dataset to 2024 and save the image of the phylogenetic tree. Now, let's move towards the question. The first question is explaining the relation between taxa. Remaining species, species 1 to species 11, are more closely related to one another, since they have a more recent common ancestor with each other than they do with species Z. Two large clusters can be distinguished in this group. First, clade species 4, species 6, species 3, and species 5, which is a larger clade. These four species are very closely related to one another, which means that they recently diverged from common ancestor. The second one is clade species 7, species 11, species 9, species 8, and species 10. This is a large clade. They are closely related to each other due to share a common ancestor and create another separate branch in the phylogenetic tree. And the last one is clade species 1 and species 2. They are closely related or known as sister taxa. The second question: which taxa indicates the most genetic changes? Species Z is taxa indicates the most genetic changes. This is why it happens because species Z is the outgroup and the branches of earliest in the phylogenetic tree. Its long branch length represents the most genetic changes compared to the other taxa, indicating the largest evolutionary difference. Question 3. Which taxa are closely related? Taxa that are closely related are species 4 and species 6, because these taxa are sharing the most recent common ancestor. If two species have a recent common ancestor, they are more related. Conversely, if they share a less recent common ancestor, they are less related. Therefore, species 4 and species 6 are closely related since these taxa converge at a common ancestor first, as we move backwards through the phylogenetic tree branches. Question 4. Which taxa is the outgroup and why? The outgroup in the phylogenetic tree is species Z. This is because species Z branches off separately and earlier than all the other taxa, which form a single clade together. In phylogenetic analysis, the outgroup is the taxon that diverges first from the rest of the group, serving as a reference point for routing the tree and determining the direction of the evolutionary change. The placement of species Z on its own distinct branch, apart from the cluster of species 1 until species 11, confirm it is the outgroup. Without a well-selected and recognized outgroup like species Z, the internal relationship of the in-group will be difficult to understand directionally, thereby leading the erroneous evolutionary conclusion. Therefore, the placement of species Z is fundamental to the structural and interpretive integrity of this phylogenetic study. And that's all. Thank you for watching.